something completely different. Here are my slides. Please present the talk. I've seen some of these talks. They're amazing. They're like the best talks you've ever seen.
They're usually fun, especially the Q&A sessions.
No,
That's a very good question. I'll pass that on to the authors when I see them.
my favorite talk is when the guy looks to the eye and John was doing some long calculations here and I have to skip over that.
Yeah. I mean, more often than other reasons is that people didn't get a visa and then
No, you can also
use this. Yeah, sure.
The visa is a good excuse though. You can just not manage to get a visa in time by not applying. This is the problem with the EU.
Of course.
The EU has completely removed this class of excuses.
I don't know what's happening. Something's eating up CPU time like crazy.
Top.
That's not helpful. Yeah, I tried. Okay, now it's better. Let's see.
At least you're making somebody some money farming Bitcoin.
Yeah, amazing. Cool. Then let's start. No, it isn't.
This declaration is it to see the hard work and
And
all of the technical difficulties that go into
the
making
glares.
science.
Yeah, the glare is so big I can't even see it. No, I didn't even have coffee. That's not gonna work. Okay, 2 more sessions, right? This is next week, and then exams start, and we could have a Q&A session, but maybe, given that we meet in the hallway often enough with most of the audience, maybe no extra session needed. There's one more topic, very different from what we've discussed so far, but somehow dear to my heart, because it's an interesting problem and has some nice combination of nifty algorithms, but also some cool biology behind it. I like it. Let's see what you say. In this next chapter, we'll look at RNA secondary structures and how to predict the structure if you're only given the sequence. And I realized that we haven't talked much about RNA, so we'll talk a bit more about the biology quickly in the usual way with some videos and pictures. And then I'll introduce formally what the problem of this prediction, structure prediction problem is and what we can do there. First of all, we've already talked about RNA in one context at least, namely in the central dogma of molecular biology. But there it was only used as kind of a boring part in the machinery to produce proteins. And here in this unit, RNA is kind of a center stage as a first-class citizen, part of the function in cells. What is RNA? It stands for ribonucleic acid. It's similar to DNA. It's formed of this chain of sugars, and the type of sugar is slightly different in the two. Which means they look almost the same often, but the RNA backbone is slightly different. It's a bit more sticky because there's one more of these hydroxyl groups that can form bonds. Whatever the chemistry in detail, this is for a different lecture. For us, what's important is that as for DNA, there's 4 different bases that usually occur and that you can use to chain together in a molecule. Unlike DNA, it's usually just a single strand. So DNA, always the 2 reverse complementary copies that then twist into the double helix. For RNA, usually just a single strand that still does twist. We'll come back to that. We've seen the messenger RNA as the key role in protein synthesis, where again, you're may remember this picture. It's a bit small here. In the nucleus of eukaryotic cells, you have DNA. There is a copy created from the DNA, and the copy consists of RNA. That's the messenger RNA, which then travels out to the ribosomes where it's translated to protein. We've talked about that in detail. Fascinating in its own right. However, this is not all that RNA does. There's many other roles that RNA serves, and there's even, I think it's not more than a hypothesis really, but the hypothesis of the RNA world, that potentially RNA was there first, and initial life forms, when life first evolved, consisted just of RNA, and kind of the more stable DNA came after. I mean, invented is the wrong term. I'm not sure. It randomly formed a bit later. RNA can otherwise serve most of the roles. It can preserve information like genes. It can have an active function in biosynthesis, and it can perform most functions that we now attribute to proteins. So in a way, it makes sense that if you had just a single building block, it might have been RNA. Okay. The video I want to show you is from some teacher training program. So it's targeted a slightly different group of people. But I think they have the nicest visuals to quickly grasp what this is about, which is 3D printed structures. Let's see.
All right, so I want you to feel really comfortable about complementarity, because that is going to come up over and over again in this module. Now, in addition to DNA base pairing with itself and RNA base pairing with DNA in order to form an RNA transcript, RNA can base pair with itself, and a DNA strand can base pair with itself. It's possible, if there are stretches of complementarity in one strand of RNA or DNA, for that strand to flip over onto itself and bind to itself. Sometimes they call that a hairpin, if it just takes it flops over and binds to itself. That isn't always the case. So all RNA is not destined to be protein, to be translated into protein. And you probably are already aware of that. You just don't maybe think of it that way. But there's a lot of RNA that is called non-coding RNA, which means it doesn't code for proteins. It is coded to be a molecular machine all by itself. It's going to have secondary and tertiary structure and act like a molecular machine, like proteins do. And you know of some of these already, okay? So if we look at this, we talk about transfer RNA. Well, transfer RNA is non-coding RNA. It's a molecular machine all by itself. We talk about the ribosome, ribosomal RNA, and that's what we have right here. Okay, it is a molecular machine made up of primarily RNA. So how does that work? So we know that proteins, based on their sequence, fold into a very specific shape that is important for them to carry out the jobs that they have, or the job that they have. And RNA is similar. It folds into these very complex shapes based on a couple of different factors. And one of them is complementarity that we were talking about before. So base pairing within itself. And if you look at a tRNA, you can see that it is just one strand of RNA that circles through and then base pairs with itself. And then what it does is it turns into a very twisted, complex 3D shape because of a stickiness of the backbone that we're not going to go into, but the fact that it's ribonucleic acid and not deoxyribonucleic nucleic acid makes it have a sticky backbone that will allow it to contort and stay into these shapes. So on this graphic, you can see that that RNA, as we know, is single-stranded. And if it is destined to be a transfer RNA and not translated into a protein, it then can base pair in this kind of cloverleaf pattern, they call it, because it's got these regions of complementarity that we were talking about before. And then, because it's got that sticky backbone, it then twists into this three-dimensional shape that is very specific for its sequence. So this structure is very specific and very required so that it fits into the ribosome just right so that it can add that amino acid to the end of the growing peptide strand, right? So that is one example of a non-coding RNA. Okay, so in addition to transfer RNA, you are also aware of of something called a ribosome, and that's what this purple structure is here. But this is.
Okay, I think that's enough for now for getting the gist of many things. I mean, I find the play area behind her really cool, where you can see how you do copies of DNA and so on. Feels like a fun thing to have. But apart from that, there was a lot of detail already in there that we'll come back to a bit. I feel seeing it in real 3D is kind of more helpful, even if it's just a video, than the animations. I tried for myself. There's certain things that in the abstract model I'll show you seem plausible, but then they don't really work in the three-dimensional reality. Okay, so apart from the functions that were mentioned, like the transfer RNA that translates from genetic code to proteins, and the ribosomes that puzzle proteins together, many other functions are known. And one key property why biologists care about the function, apart from predicting, care about the structure, apart from predicting what the function in the cells is that for RNA, often the structure is preserved among different species, but maybe the sequence isn't. So if you were to look for parts in the genome that correspond to the same function, they might, as kind of the letters in the genome, look very different, because exactly what basis you have is irrelevant if they don't change the shape. And so you can make substitutions as long as they're paired in a way that they don't change the structure. They don't really affect the function. But if you want to detect similarly working molecules in the genome, you have to, instead of just the sequence, look at the structure. Otherwise, it doesn't make sense. That's the other reason why people care about predicting the structure a lot. Okay, how do people even know how to make these 3D models? How do we know that they are somewhat realistic? There are lab techniques that you can use to get the 3D structure. I think I mentioned this very briefly when we were at the very beginning of the module, talked about protein sequencing. Remember the turnpike problem and these things. In the end there, I said the much cheaper way is to throw the molecule into a mass spectrometer, break it into random fragments, and then throw it into a mass spectrometer. If you want the actual 3D structure measured, that doesn't work, but you can clean up the protein or the RNA so that you have it in very pure form. Then it can crystallize, and these crystals you can throw in an X-ray and observe the pattern. It's a very indirect metric. It's a bit like computer tomography. So very where a lot of indirect reasoning about the structure involved. Needless to say, it's also expensive and very laborious. So you can do this for some important molecules, but people are very interested in doing cheaper alternatives. Unlike for proteins, the alternatives from the machine learning side of things don't seem to work for RNA so well yet. And the main reason is lack of data. We'll come to that a bit later again. I want to show one quick animation that shows another problem that after that video, kind of as a disclaimer, we'll ignore. And that's the fact that there's not just a single structure usually, but rather RNA.
All right, so
that we had. That's the one. So they like that. They tried to measure in real time how these molecules behave and look like, and it doesn't immediately find the right structure, but rather this yellow part forms step by step. In that initially it's in some random shape, and then step by step it forms into the final structure, the final shape that it then takes. But you see it still wobbles around, and that seems to be, let's just play it again. That seems to be also something that happens. Remember, these are all essentially in water in your cells. And it's reacting with other things. It might depend on how much salt and how much acid there is in the environment, in exactly what shape it forms. So some molecules are also known to flip between different conformations, and that also has function. We'll mostly ignore this and just focus on predicting the single most stable form. In particular, for comparing, say, between different species, which RNAs have the same function, that's good enough. What we'll also look at here, because there's not enough data to do much better, are de novo approaches, which means we're just given the sequence and out of nothing we try to predict what the structure could be. So most of the models will have to somehow just incorporate the chemical knowledge of what conformations are stable and not use much else. Questions up to this point, or roughly clear? Like, this is just vague background. I haven't said any technical specific information yet, right? Let's see. Okay, I don't think anyone's watching live online, so probably don't have to watch Slido all that much. Let's now translate what we've seen from the biology side to a mathematical model of this. And as before, we'll essentially assume the RNA is a string. It has a direction. I don't know if I stressed this all that much when we talked about DNA and genomes. The molecules have a direction. There's a distinct 5' start and a 3' end. The sugar backbone is not symmetric. So you can't just say, I'm going to read this string left to right or right to left. There's a direction. That will be somewhat more important later on. The sequence, which we think of as the input, that's the thing we can easily get because there's these high throughput sequencing techniques that can read off from, at least from DNA at low cost and high speed, lots of sequences, at least short chunks. And because RNA is directly copied from the DNA, you can get that string very easily. For DNA, you may remember that there was T for thymine instead of U for uracil. So there's a different base in RNA, but it's chemically similar. So that the name is kind of further away than the chemical structure as far as I understand. This is what we get as input. what we'd like to predict is the three-dimensional structure, but that seems hard. So instead, what we do is focus on the secondary structure. And this you should think of just some pairs. By and large, that's a good enough approximation that some of the bases in the RNA fold so that they are close together in space, and they form a hydrogen bond, a chemical bond between the bases. So the abstract representation for the three-dimensional structure, or our approximation to that, is just a set of pairs. They should be disjoint, so every base can be part of exactly one pair, or at most one pair, I guess. And there's a further restriction that they can't be too close to each other. In a way, if there's pairs here and here, you need a few in between for them to be for them to form a pair. Otherwise, the backbone would have to do too sharp a turn. Okay. We'll also require that the bases that we consider a pair are actually able to form a bond. And that depends on this, the bases that they have there. Like in DNA, there's the pairs C and G are complementary, so they're oppositely charged in a way. This is more like the hydrogen and hydroxyl groups match. They're also charged, I guess. So you could say it's two magnets. So C and G can form a bond, and A and U can form a bond. That's the same as in DNA. That's the complementarity that we had in DNA as well. But it turns out that G and U can also form a slightly weaker bond. These are often present in these structures as well. So it's a bit less clean in a way. So these 6 pairs we call the canonical base pairs. They tend to be stabilizing. If they are close together, they attract each other and they stabilize the structure. Whereas other attempts to bring other bases together, rather you get a force to push them away from each other. Okay. So first attempt, if these are stabilizing structure, one first attempt to compute a good structure for an RNA, or a likely structure for an RNA, would be to maximize the number of bases we can pair. Okay. Before I move on, can you think about what computational problem that would correspond to? So think of the backbone as this linear arrangement. Then there's all the bases lined up here. And I'm just drawing them as dots, but really, of course, they're one of the 4 letters. Okay, my example is too big. So really, this is A, C, A, G, U, A, whatever. Now I'm asking to get the maximal number of pairs possible. How would you model this? Thoughts? Yeah, it's hard to hide in this room. So one option. Sorry, say it again.
Kind of string alignment?
Yeah, maybe. Think of it more brute forcey for now. For example, every base with a C could pair with a base with a G. So for example, that's in gray, I'm drawing a hypothetical pair. Okay, this is a possible pair. I'm not committing to that yet. But then there's another G here, so I get another possible pair. I may not get a pair between this A and U because they're too close, but by and large, that's it. Right? So any other, these I would get. Now remember that the pairs have to be disjoint. So if you think about this as a graph and forget the backbone edges, then it's just a matching. And indeed, I mean, it's a maximum matching. It's not trivial to solve, but we could do it in polynomial time, even in general graphs. And so you could say problem solved. here it's even, if you ignore these minimum hairpin loops, it's even a trivial problem, because some of these pairs, in particular C and A, they only have one other base that they like to pair with. So if you greedily form all the pairs with CG and AU first, until you run out of bases for those, and then do whatever is left as GU pairs, that's going to be the best possible. Because of the minimum hairpin length, it's a little less obvious if that's the best possible. Because there's some, maybe you end up in a somehow you end up trapped with pairs that are too close. But you could just model the thing as a graph, and then all the edges that are in the graph are possible bonds, and you pick a maximum matching. Okay. Now, unfortunately, this is pretty useless. For one, that's why I spelled out this half-broken algorithm, which I guess is not okay to just ignore this requirement of the minimum hairpin length. But if you assume this, then the result just tells you the number of pairs is essentially fixed by the letters. And so there's going to be gazillions of different solutions, all with the same quality, but very different structure. And what's more, if you think about this again, a bond, a pair, means that you have to somehow contort in space the structure to be close. When the two bases are paired, they should come close together in space. If you do an unrestricted matching of this type, it might just all force the whole thing to be in one place. That's not going to work in finite dimensions. So this is, in a way, too unrestricted. We can't allow arbitrary pairs. But the thing is, any specific pair in isolation is fair to allow. Apart from this minimum distance, it's quite possible that you have some specific pair from far away. It's just that once that you have done some pairs, some others become less likely. So the problem is a bit more interesting than that. And to get a bit of a feeling what is more realistic, let's play a game. And I'll explain in a second where this comes from and why someone set out to make a game like this. What you see here is one long chain of RNA. At the moment, it's totally unpaired because it consists essentially all of A's, with a few C's and G's sprinkled in. And you can now change what it looks like by adding a few more. Yeah, the sound is very useful. And remember that C and G attract each other. So if you add enough of these, it starts to form into some different shape. Now let me add a few others. Oh, it's very slow. Poor laptop. I'm actually clicking, but okay. So you see, I've added a bunch of G's and C's that should attract each other. But if you space them out too much, they don't. Okay? You need to bring several in a row together for that to work. And now you may also notice these numbers that appear here in these regions. The negative numbers, the green ones, these are stabilizing because they're intended to mean free energy, which is how stable is the structure. The lower the free energy, the less it wiggles around and the more stable it is. So you see these huge unpaired loops here, and similar here, they have a positive contribution, which means unstable. Whereas these guys here, nested between two adjacent pairs, they're stabilizing a lot. Then there's fun things like sometimes if you put a G here, watch the number here, it's 479. And now it suddenly went down, even though I didn't close a pair. Nothing changed in the layout, but this number went down because sometimes this G and this A, they still attract each other a little bit, even though they don't form a pair. So you already see it's not always quite as clean. And you can also form AU pairs. And there's funny things happening here as well. Watch these two numbers. If I flip around this pair, they both get worse. So there's things like AU pairs, they like to be alternating, whereas the GC pairs, they like to be aligned. So there's a lot happening in the chemistry that's not shown at this level of abstraction. Okay? Fine. How are we going to make sense of this? The first order of abstraction is the stabilizing effect, by and large, comes from these adjacent pairs. They had the biggest effect. Remember at the very beginning, I put just a few green and blue and red on opposite sides, and they immediately found each other. So by and large, that's the biggest effect. Okay, what about this game? It's called Eterna. Cheap pun. It's some collaboration of CMU and Stanford. It's been running for a long time, which is impressive if you think about how short-lived some projects in science can be. It's also fairly well done for an academic project, I would say. They hired people to do the programming, I guess. What you have to do in the game is actually slightly different than what I showed you. Maybe I'll show you the actual game. In the game itself, you're given a target structure. This is what you're supposed to build. And now you have to set the basis in such a way so that this is the outcome. And oftentimes, that's the more applied use or the medical use. You know roughly what function this molecule should play in some cell. And so the structure dictates that, but you don't know how to make a chain so that it folds into exactly that structure. And the game says, maybe you can do it. It's also an attempt to make the database grow in that they crowdsource designing interesting molecules. And once in a while, when they have interesting candidates, they synthesize them in the lab and then determine the structure to then check if they can improve the models. Of course, this is a simulation. So also the structure that you see there, it's flat and everything. It's not going to be the real thing. They try to get closer to a more realistic model with the same kind of engine. It's there to design molecules that might work. And also, while you check whether they work, you get better data about the models to predict. It's quite fun to play, so if you want to help science, you can give it a go. It gets hard very quickly. What it teaches us is these two things. Number one, mostly these neighboring pairs seem to stabilize molecules. And also, it's a pretty good approximation to draw the whole molecule in the 2D plane as opposed to the three-dimensional structure that it really has. And so that's what people usually work with. I love this term I learned from a video, the roadkill diagrams, because you run over the RNA with some truck and then that's what's left. This is the same structure taken from Wikipedia, at least. I hope they're right. There's some parts here that are not actual bases, but they're labeled with the function that they fulfill. But here you see, for example, here this endpoint, right? That's the yellow and purple part. Whenever you have these pairs of neighboring bonds, they turn into something that looks suspiciously like DNA, the double helix. And in fact, that's very similar indeed what happens in DNA. Turns out the geometry is slightly bit different, but if you measure the angles and so on. But it does have this helix shape. It always turns in a certain direction. And if you have two complementary strands that run in opposite ends, they can nicely corkscrew together. And that's what comes out of these ladders that we draw flat. That's why they're stabilizing, because they tend to have a whole region forms into these twisted shapes. Okay, so roadkill diagrams and these neighboring pairs, that seems to be the thing. Adjacent pairs give the main stabilizing effect. In biology, people call these stacked pairs. Don't ask me why stacked. I guess in the 3D structure, maybe it's they slot into each other in that sense. The whole conformation of several of such stacked pairs is then called a stem. I guess it's maybe a bit more mnemonic. And again, in 3D, they look a bit like the double helix of DNA. Funnily, and I have to admit, I don't fully understand why. This seems to only work with complementary stems. So complementary strands, if one strand goes here and the other goes in the opposite direction and they're complementary. If they go parallel, it doesn't seem to work. The angles in the bases here, these are the hexagons, they don't seem to come out at the right angle. So you'll have in and out, right? This strand goes in one direction, the other goes in the other. Then they can slot together. If they both go parallel, it doesn't work. Curious, but seems to be a thing. So how can we model this? Again, let's get rid of most of the details for now and rather just maximize these stacked pairs. So here's, I guess the drawing is the clearest. Again, I'm assuming this goes in one direction and out in the other. So these are complementary and opposite directions. And then I have here a bunch of pairs. I'm only going to count pairs of pairs that are adjacent. And because it only works with complementary strands that go in opposite directions, it means if I have I and j here, I'm going to have I plus 1 and j minus 1 here. So I can count these in terms of formulas for the pairs of indices in a very simple way. So that's the second version of this problem, our second attempt. Given the structure, the sequence, let's find a structure that maximizes the number of these pair stackings. Okay. Unfortunately, that's a hard problem. It went from maximum matching, relatively easy polytime to an NP-hard problem, because you can effectively encode things like bin packing in this. I'm not going to go through the reduction, but maybe you can make it out from the example taken just from the paper. You have the bins here of 5 Gs, and then you have items of 3 and 2 and 3 and 2, separated by some A's. If there's a way to pack the bins tightly, then you get here these nice stacked pairs, but only between the elements that are from the same item. If you have to branch out to other things, then it doesn't work, and you can count that you only get the full number of stacked pairs here if you can fit the items in the bins without slack. And so you can encode bin packing, hence the problem is in general hard. You can even look a bit closer. The paper does a bit more than that, but I'll leave it with this general message. We've now found a more reasonable formalization of the problem and ran into the complexity wall. So if that's hard, how did these guys build the game where you, for fairly large instances, immediately get some prediction, right? This is they're doing this whenever you click, they predict some likely structure. Do you need a break? Shall we do a break now? Break? Okay. This again.
Who is willing to answer your question about the exercise? Which
Uh-oh.
Not an uh-oh. It's Did you make a mistake again?
I don't know, I don't think so, but I'm simply I'm not able to understand the exercise.
I think that counts as a mistake. Which exercise?
Yeah, well, which one?
Exercise 1B.
Which what does it say? Yeah. Suppose S1 plus n plus 1 contains every number from 1 to n and dollar sign exactly 1. Can that string be the result of a Burrows? For how many such strings are they the result of a Burrows-Wheeler transform?
So, wait.
I just reread the question actually.
This is one string.
Yeah.
And this n plus 1 is why?
Because I want to have n plus 1 because you have the dollar sign.
So this is the dollar sign. Okay.
Well,
The dollar sign, right? Yeah.
so no, the point is, let's say I start with a string, arbitrary string, that contains the numbers from 1 to n exactly once and a dollar sign at the end. Right?
Yes.
And I apply the Burrows Wheeler transform and it gives
Yeah, sure. Because dollar sign.
Yeah. So what's the size? What's the size of the set of possible outputs of the function Burrows-Wheeler transform of permutation of 1 to n, $10?
Yeah. Okay. That's good.
Yes.
Uh,
This is actually a trick question in fact.
the alphabet are the single digits.
What?
The alphabet.
Yes, the alphabet is every number from 1 to n.
That's the alphabet.
There's n different symbols in this alphabet.
You can, for that one, call them as you like, but you might as well use numbers.
Yes, the number, a 5-digit number is not 5 different symbols.
Yeah.
This is a, okay,
Interesting.
I just realized, Sebastian,
Yeah.
that this thing, the observation here is the same as my observation yesterday to goods.
Maybe.
Think about it. A function is an inverse,
Mm,
right?
sure. Yeah. Maybe you're overthinking this one. It's maybe a boring one,
This is a fun question.
depending on whether you already know the answer or not.
It has a one-line proof.
So, by the way, for all of these alphabets, the dollar sign is always the first character in the alphabet.
Yeah,
Okay. I mean, does it matter actually?
it's just a convention. I mean, you want the BWT to be well defined, and so you have to decide what to do. It doesn't really matter much.
Okay, for BWT it doesn't matter. For the suffix array it matters because you want shorter strings to come before longer strings, yeah?
And it matters for exercise.
Sure.
Yeah, I guess it's generally a useful convention,
Yeah, sure.
but I don't think anything substantial goes wrong. A trie, I mean, Tamio, a trie is not actually sorted, right? Or you're talking about suffix array.
Suffix array.
Sure.
You want it to be
Yeah.
less than, so that
Yeah.
shorter string come first.
Yeah, it's the natural thing to put it first. Okay, I'm in the wrong time zone here, by the way. It's only Thursday, right?
Well, I started thinking about it 1.5 hours ago.
And don't overthink. What's the stupidest idea you can come up with? And to guarantee it's correct.
And ready for the topology part of it?
I love topology.
No, there's no actual topology. But I mean, pseudo knot already sounds pretty fancy, so.
I'm very proud of myself. I have a paper on topology despite knowing no topology. The power of co-author is amazing.
Sure.
I have a paper on despite knowing no other public work. If I keep going this way, people might actually think I know.
Stupid answer.
Dangerous.
Because the Boris wheel is reversible. Can only be 1 per n.
Only one per n. The first part of your sentence was very good. What is 1 per n?
I think you just want to flesh out more precisely what you're saying.
The first one is number.
Yeah.
For the number n of how many numbers should be in the string.
How many, if the function is reversible,
Yeah.
what does Cantor tell us about the sets between the two things? The sets subtended by the function that is reversible.
For finite sets, let Cantor sleep and
Now you're confusing.
success. Mission accomplished.
Numbers, just 1.
How can it be just one?
If n is fit.
How many strings are there?
Gentlemen, shall we preserve some of the exercise discussions for the tutorial?
Yes. No.
What's that?
Well, yeah, sure.
Yeah, no, what? This is due tomorrow, right? As always.
Yeah.
Sure.
Deadline is after this.
And it's the last one.
The same format anyway. The same group of people are gonna take this.
Okay. Sure.
Okay, let's see what we can do. We found a reasonable abstract formulation for good secondary structures, but it's too hard. So what do you do if a problem is too hard? You restrict it to some special case. And indeed, in this case, it's even biologically helpful to restrict the problem a bit further. And the reason is basically in the pictures that we've seen before. If you look at these roadkill diagrams, what do you not see? Are crossing lines corresponding to the pairs or the backbone. It just runs nicely through here. And if you want to say it in other terms, it's a planar graph. And it's in a way even more restrictive. So first of all, it seems reasonable that we don't need crossings from the one example I've shown you. And indeed, it's true that most pairs that you find in real structures don't have crossings. It's also true that whenever in biology there's a rule, there's some counterexamples. So sometimes crossing structures exist, but the crossing only involves few pairs. And so we still get a reasonable information about the overall structure if we ignore the crossing pairs and focus on getting the other ones right. And it turns out the right formalization here really is an outer planar graph. If you think about the backbone as part of the graph, outer planar means there's no crossings, the edges never cross. There's a way to draw the graph so that you can align the vertices. Edges are straight lines, they never cross. Straight line is not important, but they never cross. Plus, the outer planar means all the vertices are adjacent to one face. And in this case, we can always draw it that it is the outer face, the part of the space that extends to infinity. Whereas, so I don't know, maybe let me sketch this a bit more. Here's our vertices. In an outer planar graph, I can connect these inside here. It's in principle also allowed to do more than a matching, even though we will never have that here. I can have things nested, but I can't have an edge like this. That's not allowed because it crosses another edge. And outer planar seems to be a good match for the structures that occur. And it has another good feature. It makes the whole thing tractable. The opposite, if you cannot draw a structure in the graph form like this, then it's called a pseudo-knotted structure. It's a pseudo-knot because it isn't actually a knot in the sense of topology. It's just twisted around. For example, you could have a structure like this, where you have pairs here and you have pairs here. Okay, let me draw it a bit bit more careful. Maybe you have here and here you have bonds. Then this you cannot draw as an outer planar graph. There's vertices inside here that would be cut off from the outer face, but it's still not a real knot. It's one nice line. So these are pseudo-knots, but for outer planar, we'll ignore those. So we'll focus on pseudo-knot-free structures. There's biological motivation for that as well. First of all, the structure is in 3D. If you restrict it to 2D, it's not so far away, hopefully. It's also a useful coarse graining of the structure because it focuses you on these long stems, which tend to be the most dominant feature in stabilizing the molecule anyways. This coarse graining might also, remember the video, hint you better at the pathway to get to that folding as opposed to just showing the final result. And the last point, of course, for us is useful. If we want algorithms that find a structure, then we need to pick a restriction of the problem where we can give such algorithms. So in a way, I should say this is so widely accepted that if you, in general, look for RNA secondary structures, people implicitly mean pseudo-knot-free. You have to explicitly include pseudo-knots in the language because it's so widely used and so widely accepted that you can't deal with pseudo-knots in general, that people will understand pseudo-knot-free as the default. Let's study these kind of structures a bit more. There's the way the roadkill diagrams we had before, the way also Eterna draws the molecules. Biologists give these different kind of shapes a name, and maybe it's worth to point this out once. So there's the stems that we already had, which is just these stacked pairs. They're the most important feature. Then there's HL here, this hairpins, where you turn around. These look boring because they're not paired, but for the biologists, these are the interesting regions because those tend to be the ones that stick out and that can do a function. So oftentimes, say in a tRNA, these bases in the hairpin are the ones that are highly preserved between different species because they might do the translation from genetic code to protein or amino acid. Whereas for the rest, for the stems, we don't care so much what letters make the stem as long as they stabilize it. And so often here you find mutations as long as they're so that the partner is mutated correspondingly. Then there's three other features. There's bulges, that's the B. When you have a stem interrupted on one side, you can have interior loops. That's a bulge on both sides. And the last one is this multi-loop, where you have more than one pair reachable from this. In terms of the graph, planar graph terminology, these are just the faces of the graph. Right? If you draw it like this, then the edges separate the plane into several faces. There's the outer face, and then there's these small faces of the stacked pairs. There's the bigger faces of these multi-loops, and so on and so forth. Okay. I don't care too much about these names, but if you read in biology sources, they often refer to these names. Now imagine you take this picture and you pull the backbone onto a circle. That means you pull away these, so these all need to move a bit outwards so that they follow the line here of the circle. That means this outermost stem is corresponding to these, maybe I could use some color. This stem corresponds to these pairs, right? And you can work out where the others go. In that representation, it's even more obvious that this is outer planar. That's the way people draw outer planar graphs. If you want to highlight that they're outer planar, because you see obviously all vertices are adjacent to the outer face and no crossings for the edges. I consider this as an intermediate stage because now if you have the circle, it's not actually closed, right? Because this is a linear structure. Let's take the circle and pull it straight. Then you get to this picture. Again, the same structure. And now you have these arcs on top. And again, it's outer planar if and only if you could draw it as a line with non-intersecting arcs all on one side of the line. And hopefully you can visually see how these all correspond to the same structure. That brings me to the last representation, which looks very innocent. Bloop, down there it is. From this linear arrangement, very easy to see. You can represent edges by opening and closing parentheses, and the other bases just as dots for unpaired. And then this string of opening, closing, and dot, that's completely describing the structure. So if you want to work in a computer with these structures, clearly that's the way to go, right? Then it's just a string, much less hassle than dealing with these pictures. But biologists, of course, prefer those because they are closer to the structure in three-dimensional space. Yeah, also note that precisely the fact that these arcs are all above the line and don't cross means that you just have to find the corresponding parenthesis. The matching parenthesis is your partner in the matching. Okay. Now you could do all sorts of combinatorics with this. You could say we can count how many structures there are. You could find out that while there's clearly 4 to the n different sequences, there's a good deal fewer secondary structures. So a good hint that there's always going to be exponentially many sequences for the same structure, and so on and so forth. We're not going to go there, even though it's fun stuff. Let's come back to algorithms and try to predict a good structure, solving our original problem. Funnily enough, the first algorithm that was somewhat useful for RNA structure prediction, Nusinov's algorithm, did what I originally said is useless, namely it just maximizes the number of pairs. But it maximizes the number of pairs in pseudo-knot-free structures, so these outer planar graphs. And there, of course, you can still solve maximum matching in polynomial time, but it's restricted to the graph being outer planar. So I don't know if there's an obvious second algorithm apart from the one that I'm going to show you. I will come back to the counting pair stackings because that was the more realistic thing. But I think historically that's correct to say that the algorithm first just counted the number of valid pairs. Any thoughts how you would do this? So think about what you're given as input is the sequence of bases, just the letters. And you're trying to find the best such balanced parenthesis string, and you're maximizing the number of parentheses. How could you possibly get started here? Or what kind of algorithm design pattern might be useful? I'm already hinting at that it's going to be polynomial time, otherwise we wouldn't be talking about it further. I mean, whenever a problem looks harder than greedy, it's probably going to be dynamic programming, no? And indeed, the good thing about these balanced parenthesis strings is, suppose I somehow figured out that this pair of parentheses is matching, and I want to have this one. Right, so there's this pair. What I do inside here and what I do outside here, it's going to be independent. There can't be any crossings. So whatever pairs the maximum number of pairs I can throw into this pink part that crosses the arc I decided to put, it's going to be independent of what I do inside here because they can't cross. So there's a natural decomposition. In a bit more structured terms, we'll come to that. decomposability is the key thing. For a dynamic program, I guess from how I showed it, it's already suggestive that you want to have subsequences or substrings as your subproblems. So formally, we'll have Dij to be the maximal number of valid pairs that you can possibly squeeze in any pseudo-nut-free structure for this subrange of your RNA. And by solving this for the whole thing, we'll make our way to the solution for R0n. And if you think about how, so I guess the picture is drawn in a way like you add one more base or so. If you have a range I to j, what could the inner part look like? One option is there is a pair ij, and there's maybe a pair inside, but certainly I and j are paired, or I could be unpaired, or j could be unpaired, and then whatever comes inside, recursively resolve it. Or we have some cut point where everything to the left and everything to the right is a separate structure that don't interfere. That's all options. In a way, if you want to see why it's exhaustive, you look at what I and j do. Either they're paired with each other, or one of them is not paired at all, or they're both paired but not with each other. Right? I think this is a bit misleading that I is paired with essentially k minus 1 and j with k plus 1 or so. This is not needed. There's just, there could be a longer stretch, right? I and j are paired with something. So there's going to be some part here, and then there's an arbitrarily long part in between. That doesn't matter as long as I can find one cut point k. So in the recursive programming recurrence, I have the number of pairs I can form is nothing, 0, if I'm too close, if I'm on a tiny piece of the RNA. And otherwise, it's the best among those 4 options. For the first one, I have to check if I can form the pair between I and j. And note, I stole the pictures from one of the books I mentioned on the website where they have I and j inclusive. But to be consistent with the rest of the module, I used here R I inclusive, j exclusive. So this is the pair we would want to make is between Ri and Rj minus 1. I can add another pair if these are in the canonical base pairs, one of the 6 allowed combinations. Otherwise, no. Then I go to the recursively computed structure between those. And for this bifurcation part, I have a maximization over all possible cut points. So clearly, quadratic space is sufficient to store all the subproblems. And because of this maximization here, I have another linear factor in the running time if I don't try to be smarter. Because this algorithm and versions of it are so much used in practice, people tried a lot of tricks to speed up, at least for typical cases, what happens. And oftentimes you can remember what the cut points were in the smaller structures and keep a list of those. So you may not have to iterate through all of them each time. There's a few tricks you can play. But by and large, you're stuck with some higher polynomial running times. So this is indeed tricky to do with very large molecules. Okay, what's left is, number 1, to get this to the more realistic model of counting pair stackings instead of pairs. I have a separate slide, but I think it's actually easier to do with these pictures. It's not at all difficult to count the pair stackings instead of pairs, because all you have to check is whether there's a way to close a pair here and also have this pair there. Now, the way I set up the recursion here, we don't know in the substructure what we did. So we have to remember an extra bit, if you like, whether we close the outermost I, j minus 1, whether those are paired or not. But if you know that, then you could say one option is I close the pair from I to j, and the one just inside also needs to be closed. That's one option. Then I can add one stacking. And in all other options, I don't get a +1. Okay, I don't know. Maybe that wasn't so clear. I'll describe the more general algorithm again, but with a more graphical syntax, and then we'll leave it at that. The maximum matching, unlike the general case, for outer planar or pseudo-knot-free structures already gives you a reasonable structure. It's much better than this original one where the numbers were essentially predetermined and everything turns into a clump. Here, this is sensible, but people observed that it's still counting the total number of pairs is not as good as counting pair stackings. And we can extend the dynamic programming. I was waving my hands already on how to do that. For going further and for the extensions that people then further did, they often went to a more pictographic way of showing DP recursions. And I think this is just, in a way, beautiful in its own right. So maybe it's useful to just show you this as an example. Maybe it helps you think about such problems. The specific recursion, maybe don't worry about that for now. Just the syntax is you're looking at a substructure from some position to some other position. It may have some constraints, like in this case, the wiggly thing means there has to be a pair. So the outermost two elements are paired, whereas what's inside here, we don't know. And so we say the value of this substructure is the best of, so it's minimization here we use to maximize. Minimum would be for free energy. It's the best of certain possible options you have. Either it could be that there's no further pairs in this range. The white area means no more pairs. Or it could be there is a pair, then you find the outermost inner pair, the rest is white, and here you recurse. This obviously has the same shape as this, so you know what to do. Right? In a DP recurrence, you would have some, this would have some name and some parameters ij, and then this would be the same name with appropriately adjusted I and j, so some new parameters I prime, j prime. Or maybe you say there's a cut point k, where these dashed lines mean I don't commit yet to what's happening here between the outermost pairs. Could be anything. Yeah, it could be paired or not. But I know that the substructure, so this part is white, which means there's more pairs in here or more pairs in here maybe, but nothing between them. Okay, this is roughly clear. And with that, you can express what I tried to describe earlier. We need two mutual recursive constructions. We need one where we know that the outermost bases are paired, and one where the outermost bases are not paired, or we don't know. It doesn't actually matter because we determine the best possible structure. If I had the choice to add another pair, that's always going to be better. So I think it doesn't matter if you restrict, if you here say the semantic is there cannot be the pair ij, or whether you say it's not fixed. It could be there or could not be there. I think it doesn't matter. Okay, so you could do counting the number of stackings in the following way. If you know the outermost pair is there, either there's nothing further in there, or you find the next inside, or you do the bifurcation. And for this part, you have the option to put the pair there and recurse, or have unpaired I or j, or further bifurcate. It's not an ambiguous decomposition, but it's good enough for a minimization. Right? You see here that if you have several ways to bifurcate, you may do this in any order, and it's not unique, but it doesn't matter for minimization. Okay. This complexity doesn't change by this. if you implement it well. Now remember what this looked like here. There were all these lovely numbers shown. Where do these come from? Obviously, they're not simply counting pairs, etc., but rather they have a chemical model in mind that's known as the Turner model, because this guy owns some lab and they've done tons of experiments to figure out for all sorts of small substructures what their contribution to the overall stability of a molecule is. So even counting these stackings is still a relatively rough approximation. It depends on the exact basis. So the bonds between GC are much stronger than AU, and GU is even weaker. Also, I showed this in the tool earlier, that sometimes adding another base in the unpaired part changes the contribution to stabilization. And even what's in the hairpin loop can influence things. So as a culmination of a lot of lab work, they published for all sorts of structures the corresponding energy contributions. Long, big tables that we don't want to go through. I just want to conceptually show how they work. And I think it maps nicely to, again, the graph representation. In a way, you could say they give an energy contribution to each of these faces in the planar graph. So for example, one for this blue part, which would be a destabilization because there's a bulge, and the green one is a stacked pair, so that would be stabilizing. And they have lots of tables that tell you, depending on what the bases there are and how big the bulges are and how big these multi-loops are and all for various combinations of things, they tell you what the energy value is, the contribution to free energy. So in theory, all you have to do is decompose a large structure into all these possible faces. If you do this directly, it actually doesn't work, because think about this huge face here where I put the text in. You could have as many pairs inside there as you want. And if you want to optimize where they need to go in the dynamic programming, these are a lot of variables. So that would drive up the running time essentially beyond polynomial. So what people do is you restrict it to 2 pairs inside. So you have this wiggly line outside, and you allow 2 more wiggly lines inside. If it's more than that, you do some heuristic and say, I'm splitting off 1, and there's an additive function that you can deal with without specifying them already. And I think the energy models also aren't so precise for these big structures. They only work well for the small faces. So with that, you get essentially the same kind of recursion as you had here. Instead of having more of these inside, you would use this rule and give a general contribution to this outer loop. So that's called Zucker's algorithm. One of the other people involved also with the papers here. But it's by and large the same dynamic programming algorithm. My understanding is that's essentially also what Eterna uses. So this is still state of the art, even though the papers are 20 years old. That brings me to the last part that I'm going to quickly teaser and maybe finish next week. What has happened beyond this? The chemistry-based approaches at some point hit a wall in that you can't get much further with refining the energy models, it seems, and also the algorithms would get expensive if you were to include more of these inner loops. So people instead tried to look towards more learning approaches. And so the key challenge is to have enough data to do that. And unlike for proteins, it seems there's not enough reliable RNA structure data to just apply some black box learning machinery. This seems for AlphaFold, there was, I think, several hundreds of thousands of protein structures are known, all like laboriously produced by some PhD students. That seemed to have been enough to train a system. For RNA, that's not yet the case. What you can do is still a machine learning driven approach, but by encoding, in a sense, more domain knowledge in the models, you can reduce the number of parameters to a point where the available data is enough to, at least in that model, get a good fit. I think apart from the machine learning side, which I'm not going to go into in full detail, there's a few cute algorithms and connections to formal languages that are fun to explore. And that comes for essentially the model that gives you this combination of a learned approach and encoding some domain knowledge. And what we'll use for that are context-free grammars and then probabilistic context-free grammars. So let's briefly remember what context-free grammars are. This is stuff you would usually do in some theoretical computer science class without any application, maybe, apart from parsing computer programming languages. A context-free grammar has these 4 components: non-terminals, terminals, rules, and a start symbol. And here's an example that's a typical one that you would study there. You can, I should have done these in green. The parentheses are part of the terminals. So this grammar, if you use as start symbol E, produces all correct arithmetic expressions where you have addition and multiplication. And you can generate the 2 variables x and y, fancy. You can generate numbers, either a single 0 or it's starting with some digit and then continuing with potentially more digits. So you can produce numbers. I don't know. You could have something like this. That would be one valid string that you can generate from this grammar. The way grammars work is that you start, how do you get to this? You start with the start symbol. You pick one of the rules to replace a non-terminal by the right-hand side of what you have here. So here we would pick this one. So you generate do it a bit more stretched out. So I've chosen this first rule and replaced the non-terminal by these elements on the right-hand side. Now I have further non-terminals, so I have to continue replacing them again using some rules. Let's replace this one, and here we use the rule that replaces this by I. And then this becomes 55, so I is replaced by C. I'm seriously running out of space. Let's cheat a little bit. Maybe I have to compact it a bit. So it goes to Z. And then from C, we go to a 5 and C prime. And then the C prime goes to 5. Then I've produced the 55 plus, so you can read it left to right. And there's going to be another subtree for this E that I don't have space for. Yeah, I know. Luckily, we're not in formal languages, so yeah, I can just appeal to you already knowing this. One way to draw how you derive things in such a grammar is this parse tree, where you have a subtree for each non-terminal where you replace step by step using one rule the non-terminal by the right-hand sides and keep going until you only have terminals. Another way to write it is using leftmost derivations, where you have a string of terminals and non-terminals, and in each step you replace the leftmost non-terminal by some right-hand side. The usual application people have in mind for this is programming languages and parsing. So apart from just checking whether you can generate a string, you can build this parse tree and then continue working on that, for example, to compile some higher level programming language into some assembly code. Here we're going to do something different. We're going to treat the grammar as a probabilistic model or a class of probabilistic models. You should think of this as a generalization of something like Markov chains. So maybe I also should have gone on the slide. A Markov chain is a finite automaton with probabilities between the states. And depending on the model, it might generate a string, or if you think about information theory, a Markov source might generate a string where the probability to emit a certain symbol depends on the state you're currently in. And whenever you've spat out a symbol, you make a transition to a new state, or there might be a self loop to stay where you are. But essentially, this is the equivalent of finite automata in regular languages, whereas the probabilistic context-free grammars would be the equivalent of context-free grammar or context-free languages. So they're a bit more powerful. Every Markov chain is also a probabilistic context-free grammar as a special case, but you can do more expressive models with these context-free models. Formally, for now, all we do is assign for each of these rules a certain probability. I guess I was a bit sloppy here as well. I didn't explain what this vertical bar means. These are 3 rules for E. All right, E to the first part, E can go to this multiplication part, or E can go to I. These are 3 separate rules in a formal sense. Each of them gets a probability, and I want them to sum to 1 for each non-terminal. So whenever I have a non-terminal, say E, I can throw some random process that determines me which right-hand side I'm supposed to replace it by. And I do this for all the non-terminals. Maybe for some, okay, here they're all at least 2 different options. Sometimes there's not even a choice, then that's fine. The important bit is that it's a probability distribution for the right-hand sides, given that you know the left-hand side. If you do that, then the probability of a derivation or a parse tree is just the product of all the used probabilities, of all the used rule probabilities. So you could say I'm starting with the start symbol. That needs to be replaced by some right-hand side. So I'm going to flip a coin that decides which right-hand side to use. That's probably going to add more non-terminals. So again, I'm going to flip a coin to replace, say, the leftmost of those. And I keep going until all non-terminals are gone and replaced by terminals. Unless I made a mistake and made it more likely to keep adding more non-terminals than terminating. In principle, I can do this, which might mean my model is stupid and broken. You can avoid this with training in the right way, though. Yeah, maybe we'll leave it at that. And I connect it to RNA next time. The 3 minutes might be a bit short for that. Okay.